Our next speaker is Dr. Junichi Takagi. Dr. Takagi is professor in the Laboratory for Protein Synthesis and Expression at the Institute for Protein Research of Osaka University in Japan. Dr. Takagi is a member of the board of the Protein Science Society of Japan. His research interests include the structural and functional analyses on receptor-ligand interactions, particularly in the field of immunology and neuroscience. And his talk today will be the Lasso-grafted Fc design platform offers creation of novel receptor agonists and antagonists. Please welcome Dr. Takagi.
Thank you, Ken. Can you hear me? No? Can you hear me? Oh, this one? Is this on? Okay. Okay. Thank you. So I'm really glad to be here back in Boston. So I have been in Tim's lab for 6 or so years. That was almost 20 years ago. But every time I come back to Boston, it comes back. The air and environment comes into me, and then that energizes me, which is very good. I'm really glad. And also, I'm very glad to be here, to be one of the speakers of this very important symposia celebrating the surfacing of the IPI at the moment, and then also can communicate with many old friends. So yes, so this is, so here we begin. So this is the title of my talk. You might be wondering what lasso grafting is. I'll tell you in a minute. But my talk probably, interestingly, have a lot of parallel with the former speakers, Yvonne and Andreas, because I'll be talking about structure and structure aspects of the signaling of the receptors. But it's probably, I don't need to tell you about how important it is to have a structure insight about a mechanism of cell signaling, particularly in a case of the single-pass transmembrane receptors. And all of us, I guess, know that most of the time these receptors are activated on cell surface by oligomerization, mostly a dimerization, sometimes trimerization. But it's not just putting this together or cross-linking randomly. It's always necessary for you to cluster them in a particular orientation and particular direction. So like Andreas addressed already, the geometry matters a lot, right? And that, I think that has to to do with the fact that, like Yvonne addressed, that the interface matters because after all, it's the interface. Which interface of the protein that you use for this heterologous or homomeric interactions that bring in a complex in certain conformation. But then saying that, then probably it's not necessarily the absolute requirement to have a natural or physiological ligand to send a signal. If the key thing is to bring the 2 or 3 receptors together in a certain orientation, maybe artificial agents can do that. That also been addressed very nicely by Andreas' talk. So, but then when do you think about what would be the ideal design for artificial receptor dimerizers? I'm just now focusing on dimerization only. But then what's the ideal scaffold? So immediately you think about, of course, antibodies, because antibody has 2 arms and which is perfect to bring 2 molecules together and then cross-link. But it's not that efficient or that very high chance that you get agonistic antibody because, as you know, two Fab arms are too flexible or too far apart to have a certain geometry of activated or signaling competent receptor dimer. So then what do you do? So these days I work on a macrocyclic peptide very often. And then the thing is that even though this is, you can synthesize this, but it's not, unlike the small molecule, it has a footprint of as large as a typical antibody has. So it has a high affinity and high complementarity, meaning high specificity. But this is just a monomeric binding. But you can easily cross-link, dimerize the two moieties by connecting them with the flexible linker, like PEG linkers. And then you can even play with the distance between the two active motifs to make a certain distance of the receptor dimerization. But in this case, too, is not enough because it's obvious that you cannot be these cannot be really rigid dimerizers because of the flexible linker. So still cannot control the geometry or topology. So what is ideal, I thought? So ideal thing probably is to have first a rigid base protein. It doesn't have to be protein, but I prefer protein. A rigid entity, rigid structure with 2 or 3 receptor binding active site moieties. And then you can probably, if you add it on a cell surface, the cell surface receptor can be clustered in a particular orientation at a particular distance. Then it can send a signal, right? So this is the ideal platform for making or designing artificial receptor dimerizing agonistic protein. But how do you make such artificial proteins? Out of the blue, you cannot just design such things. That's where it comes to our technology called lasso-grafting that we developed several years ago. Together with Hiro Suga at Tokyo University. So he is a peptide chemist, but he's famous in developing this, what he calls rapid system, which is a macrocyclic peptide discovery system. So by way of recoding the genetic program, he was successfully introducing the unnatural amino acid. So in this case, chloroacetyl tryptophan or tyrosine will be on initiation codon. So it's an N-terminal. And then because of this chemical moiety, it can spontaneously cyclizes, making a thioether bond with the C-terminally placed cysteine residues. So all of and then of course the internal sequence is randomized. So you have a large collection, a highly, highly diverse library of a macrocyclic peptide or display system that always make this macrocyclic structure. And then what you do is regular panning type of the selection. So in the end, what you get is this cyclic peptide with thioether bond. Of course, you have a different sequence of the internal sequence. But when you have this selected this peptide against your target molecule, it can be receptor or other molecules. And then what you see if you look at this structure is the common unit. The cyclization unit is identical for all peptide, but of course the internal sequence, which will be the binding unit toward the target molecule, is different. And of course, if you look at analyze the structure of the complex between these, some of these peptide and the target, Yes, the shape of the peptides are a wide variety. There's a helices or a beta strand or a random coil, but almost always this cyclization unit, the tri-ether bond, is facing away from the protein. So what you need actually is this yellow part only. So in a way, you don't need this cyclization unit. But of course, you do need this to make it cyclic and then fix the conformation. But for the contact or the recognition of the target, you don't need this. So then I spoke with him. Maybe we don't need this chemical moiety because we can replace this thioether bond. It can be disulfide bond, but this replace this chemical bondage with protein. Okay, because protein has lots of protruding loop on a surface exposed loop. And then all we have to do probably is to find the right place to insert this internal sequence, making just the protruding loop, single loop, sticking out from a certain place in your scaffold or base protein. And then this should gain a binding specificity that was originally implemented within this cyclic peptide. So you may think this is too simple. It doesn't work that way. I thought so too, actually. I was, not really expecting this to be universally applicable. But finally, we found out, probably because of the highly optimized conformation and the sequence of the rapid selection-derived peptide, this works beautifully with very high success rate. And then in terms of the base or scaffold protein, it can be almost anything. You can insert your peptide sequence into the loop, multiple loops in, say, serum albumin, for example, or IgG, not at the CDR, but any other places, and including other enzymes or single domain proteins. So it's very robust system. Again, so you have many cyclic peptide binding to different receptors and so on. And then it can be implanted onto a different scaffold. And then you can choose the scaffold. So, and then this tells you that this technology is highly transplantable or mobile, meaning that once you have this peptide sequence that you want to insert into a protein, that can be moved around within your protein or onto a different protein. So that means it's highly mobile, transplantable, and which is very in contrast with other existing technologies. So usually, of course, antibody has 6 loops, CTL loops. The nanobody has 3 loops, and mostly these binders or artificial binders are selected because of the collaboration with the multiple loops. So even though you can have a very nice high affinity and highly selective binders, what you get in the end is the sort of 3-dimensional pharmacophore structure, not the sequence. So if it is made of 3 loops together, it's very hard or almost impossible to implant this set of the pharmacophore onto a completely different protein surface. In contrast, in our case, it's just a single loop because it started from just a single cyclic peptide, right? So it's a single loop. So that's why it can be transplanted onto one protein at one site, or the same sequence, same active moiety can be moved around within this protein or onto the different surface of the protein. And if you wish, at multiple times, like so. So that is a difference, I think, of our technology with other existing technology. So now, like I said, almost all of the protein that you can think of can be the base protein for this, or scaffold protein. But our favorite one is, of course, the antibody. Of course, we know that the tip of the Fab is used for engaging antigen binding. So other than that, you can actually manipulate. So in a type of the molecule that we call antibody, we can just leave the Fab CDR loop alone, but implant these rapid peptide-derived sequence onto the different portion, particularly at the tip of the, or base, or in the middle of the Fc region, okay? And then in an instant, you will have bispecific, trispecific, even tetraspecific antibody without hassle. And also, so then I realized that maybe we don't need Fab fragment, because we can implant the binding specificity by using this technology. So we don't need the original binding. So these days, my favorite scaffold is just Fc-only protein, okay? And we decided to call this mirror body, the miracle body. I know it's a childish naming, but I like this. So, but anyway, we found that there's at least 8 different locations that you can insert your peptide, the top region, middle region, and bottom region. So you can move around your binding moiety, binding sequence onto a different sites, but if you just choose one, that's already a divalent binder, because Fc is a homodimeric molecule. So, but how does it work? Well, it works, like I said, like nothing. Here I have 7 different peptide sequences already selected against a particular target antigen, like these, and then implanted into the same site of the Fc, what we call B2 site, and then ending up with a collection of Fc-sized, almost Fc-like molecule, right? And we can use these mirror bodies, I call, as a regular, like as if it's a regular monoclonal antibody. You just stain the cells. These are the transiently expressed cell line with these receptors. And then you can use this reagent as a monoclonal. And you can use actually plain secondary antibody against human Fc. And then you get these nice staining. So it's very, but still, it's not antibody. It's about 50 kilodalton Fc-sized molecule. So meaning there's no light chain. It's a single subunit entity. And it's very convenient. So, and this is the shape of the what I call mirror body. And then in reality, in real structure, it looks more or less something like this. So as you know, Fc portion is a rather rigid molecule because of the very strong association with the CH3-TH3 interaction here. And then also, in addition, you have this cyclic peptide derived sequence inserted into a certain loop, having this. So this is, like I said before, could be the ideal base protein for making receptor dimerizers, having a rigid base protein and 2 receptor binding moieties. And then it should be able to cross-link 2 receptors together. If you design this in an appropriate way, it will send signal. Okay, so let's prove that in a real case. So first case was the MET or c-MET receptor. That's a tyrosine kinase receptor activated by HGF, hepatocyte growth factor. And HGF will engage two receptors and then send a signal, create autophosphorylation, and then send a downstream signal. And then HGF is a very, or a CMET signaling is a very important signaling process. And then it's responsible for example, hepatocyte regeneration and a branching formation of the epithelia, and even the survival of the neurons and so on. So in theory, HGF can be a great biotherapeutics when you apply this. But unfortunately, HGF is not a very good viable biotherapeutics at the moment. Why? Because it has a very short plasma half-life. It's very unstable. It probably sticks to everywhere, and it's lost rapidly. So our goal for this particular project was to design a MET dimerizing unit like HGF, but with a very long half-life in vivo. So how do we make HGF surrogate or receptor mimic MET agonist out of the Fc protein? Luckily, we already had a rapid peptide system selected against CMET. We already published a while ago that this cyclic peptide binds strongly to human MET receptor. Okay, so we already had a sequence to implant onto this scaffold. So what we do is very simple, using 8 different insertion points that we found out that was compatible with this technology, made 8 different versions of AMD4 derived mirror bodies. So all of them have an identical sort of amino acid sequence or a composition, only the position of the peptide insertion was different. So, and then we expressed this and purified it and added to the MET expressing cells, see what happens. And instantaneously, we learned that, yes, we can get agonists. So AMD4 inserted mirror bodies, the Fc-only protein, depending on the position of the insertion differently, but most of them actually could induce phosphorylation of MET. So it can activate MET on cell surface. And it was interesting because depending on the site of insertion, it has a different concentration dependency. But when we measure the apparent affinity or effective affinity of these mirror bodies, remember this is dimeric, so it's not the real KD value or anything, but just apparent affinity of these 8 different versions. One actually it did not show quite well binding, but others showed binding, but with the different affinities. And if you compare this table with these figure, you will know that the agonistic activity, the strength of the agonistic activity is roughly correlated with the affinity. So it binds stronger, then it sends signal more, okay? In this case, that was the case. So please remember this because I'll come back later with the different type of the mode. So anyway, because our initial purpose was to make it as a HGF surrogate, so what is the effect? It's not just phosphorylation. So this AMD4 mirror body, even though it has a completely different structure or sequence with HGF itself, but it induces the same set of genes using a human hepatocyte culture, or by using a humanized liver mice, it can actually stimulate the hepatocyte growth in vivo when administered directly. And the more importantly, it has a very long plasma half-life because it's basically Fc protein. So it can also bind to FcRn, the recycling receptors and so on. So it has a very long plasma half-life, about 50 hours. almost identical to the plain FC. And if you compare it with the real agonist, HGF, less than 30 minutes, it goes down. So it's a very long-lived HGF surrogate that can be administered, at least in mice. So this can be a very nice, there's a potential that it can be used as a biotherapeutics, again, liver diseases like NASH. Okay, that's the case for MET, tyrosine kinase receptors. What about other system? So now we turn to a different, completely different receptor system, which is plexins. So Yvonne already nicely addressed the importance of this class of the receptors. It's a general receptor, signaling receptor for semaphorin ligand. And particularly, but here, what's important to remember is that plexin is also activated and sends signal by dimerization. So because, like Yvonne told, semaphorin is always a homodimer, so it automatically, by engaging plexin, it cross-links two plexins together. And in this case, it's not a tyrosine kinase. It's a gap domain that exists at the cell cytoplasmic tail, and then it gets signal on. So again, if you can cluster or dimerize plexin in an appropriate manner, you should be able to get artificial agonist. Okay, so we chose particularly this system, plexin B1, which is a signaling receptor for the molecule called semaphorin 4D, because we already have isolated a couple of different cyclic peptide that binds to this receptor with a very high affinity. So this has been already published. So we decided to use, to begin with this peptide, what we call M7. So again, this is the sequence. It's a cyclic peptide, but the internal sequence is linear, like this. So we inserted, like the way we did for the MET binders, just made 8 different versions of mirror bodies, the FC-only proteins, and then express them and purify them, and then add it to the cell that expresses plexin B1. See what happens. So this is the result. So when the plexin gets signal activated and sends signal, then cell basically gets shrink. So this is the impedance type of the assay. So when you have activation of the plexin, you see what we call cell index goes down like this. So this means that this one was activating plexin B1 on cell surface. So as you can see here, it's interesting. Not only M7 inserted mirror bodies, the FC-only protein, can send signal, but it's highly dependent on insertion site. So out of the 8, all of them actually binds well to the cell surface plexin. So almost no difference in terms of binding affinity. So they should be cross-linking two plexin B1 receptors together in a same, almost similar efficiency. But only this one, well, at least compared to this one sends signal the best, okay? So it's highly topology or geometry dependent. So in this case, unlike in the case of the MET mirror body, the activity, the agonistic activity is not governed by affinity, but rather it's governed by something else. Okay, but also when you talk about the artificial agent like this, usually that has a much less activity compared to the real ligand, physiological ligand. But in this case, this is the concentration dependency of the most active M7 mirror body. It has a EC50 of 3.3 nanomolar. Compared to the real ligand, the SEMA4D, it's actually the same. So it was as effective as the natural ligand, at least in this case, in this type of assay. So it's not that bad. So, but then what about other peptides? So this is just one peptide. What about other peptides? Like I said, we have isolated already different class of macrocyclic peptide. In this case, I'm talking about this M6A9. M6 class of peptide, cyclic peptide, had actually a stronger affinity to this receptor. So we just did the same thing, inserted this sequence into the 8 different positions in the Fc, and then obtained these 8 different versions. See what happens. So when we add this to the cell culture system, like before, nothing really happened. So this means, in this case, in this particular peptide inserted mirror body, they cannot signal at all, okay? Although all of them actually can bind to the cell surface plexin rather strongly. Okay, but it's not just that. When you add, after this pre-incubation, when you add the true ligand, SEMA4D, surprisingly, some of them actually strongly inhibited the signaling. So it's not agonist, it's antagonist. And also we know as a fact, it's not competitive antagonist. It's something else. And also the effect of this inhibition was highly site dependent, meaning it's topology or geometry dependent. Okay, and then in this case, the strongest inhibitor in this case was the B3 inserted version. So why do we have, so this is another side story. This M6A9 mirror body is being tested by our collaborator, Enrico Giraud and Luca Tamagnon at Torino, who are interested in breast cancer. And then this antagonist of plexin B1 somehow suppresses the tumor growth in this mouse triple-negative breast cancer model. And then we know as a fact, it's not directly acting on plexin B1 expressed on a cell surface of the tumor itself. It's actually affecting the tumor microenvironment, primarily CD8 plus T cells, which is very interesting. But anyway, this can be, I mean, the plexin B1 antagonist can be biotherapeutics. That's the point I wanted to make here. But then why is the case? Why one mirror body, the FC molecule, strongly activate plexin B1, while the other one is a strong antagonist? And the strongest version is B2, B3 inserted version. So it's right next to it. So globally, they look very similar, right? So why this is antagonist, this is antagonist, this is agonist? And maybe you have already guessed it's because of the topology. So we have to solve the structure of the complex between these peptide and the plexin B1. Yes, we did that. So actually this M7 and M6 peptide bound onto the same, what we call SEMA domain of plexin B1 at the N terminal, the same domain, but on a completely different surface. It's like a back and front. So it's on a separate, completely different surface. So the interaction surface actually governs the overall topology for sure. So we don't have the actual structure of the entire plexin B1 domain, but we have a pretty good guess about the overall shape of this receptor. So if we can, using these information together with the previously solved structure of the receptor-ligand complex, we can model what will be the expected shape of the receptor dimer that is activated by agonistic or antagonistic mirror bodies. And this is the answer. So when the SEMA40, the true physiological ligand engages, it forms something we call face-to-face dimer, and as well as this M7 agonistic mirror bodies. And if we extrapolate the entire cell ectodomain of the plexin B1, it looks something like this. So these two cases, maybe it's slightly different, but overall it's very similar. They have a similar face-to-face dimer, and which is ideal for the sending a signal, because the two transmembrane domain connecting to the gap domain at the cytoplasmic phase is close together, as opposed to this, antagonistic M6-based mirror body. Even though we try to, manipulate this really hard, there's no way that this engaged dimer have a close opposition of the gap domain. So that's why this is antagonist. So meaning that it's not antagonist because it outcompetes with the ligand, but rather it actually fixes the inactive or signaling incompetent conformation of the receptor. That's why it's strongly inactivating. So I think this is a very interesting mode of inhibition. If you think about, shutting down the signaling of certain signaling receptor, this could be another route rather than, trying to inhibit the ligand binding, but, forcing the inactive receptor dimer. And like I said before, our mirror body, the Fc-sized, rather rigid, divalent molecule, will be a very good platform molecule or molecular shape that you can explore this type of either agonistic or antagonistic topology-defining, geometry-defining artificial agent. So in a few slides, I'm going to go over, these are the already published results. The EGF stuff is unpublished, but it's, I'm going to share it with you. So again, Andreas already told us about the general EGF receptor signaling mechanism. It's very famous. Everybody knows that. And then in general, EGF receptor is on a tucked, compact, inactive conformation, and after the ligand binding, it dimerizes and sends a signal. But in general, also, it's known that there's some intermediate shapes or states exist. That is maybe conformationally changed monomer or signaling incompetent preformed dimer that exists. But it's the ligand binding that pushes this equilibrium forward to the active receptor. That is, I think, a generally agreed signaling mode of all of the EGF receptor. So again, we already have somehow this peptide that binds strongly to the EGF receptor ectodomain. So we try to do the same thing. Again, the slide is almost identical to the a couple of slides I already showed. Just use this peptide and insert it at 8 different places in a FC portion, and add it to the cell surface. Yes, 8 different mirror bodies could recognize cell surface EGF receptor nicely, except for this one, actually, this T2 version. was very weak binder somehow. We don't know why. So except for this one, all of them recognize the cell surface EGF receptor, okay? So it should be dimerizing the receptor on a cell surface to the same, almost same extent. But when we measure the activation of the phosphorylation of the EGF receptor, then we realize that it's, again, site-dependent. So among the 7 binding mirror bodies, 8 actually, one did not bind, but I'm including this one. But anyway, the B1 based or B1 inserted mirror body could activate the strongest. So it's clear that it's compatible with the active receptor dimer conformation. So yes, just clumping 2 EGF receptor together on cell surface is not enough. In this case too, the topology is important, as we already know. But from this different approach, we can also show that geometry matters. Okay, so that is it. And then I'm just thanking all the people who did this work. Katsuya Sakai at Kanazawa University, he was the main person who did the MET agonistic project. And Nozomi actually did most of the work in the Plexin B1. And the graduate student Azusa is right now doing EGF receptor stuff. And I think all the collaborators, particularly Hirosuga at Tokyo University, who is essential for carrying out all these technology. And I thank you for listening. Questions?
It's a very nice talk. The grafting these peptides in many proteins, it will create a new epitope, so B cell epitope. And don't you think that will relate to some antibodies against these grafted proteins that will clear from the system? Maybe 1 or 2 time injection is okay, but if you do repeated injection, you may get antibodies against the grafted proteins. Don't you think that's a concern?
Are you asking about the potential antigenicity of the
Yes,
peptide
yes,
portion?
yes, exactly.
Yeah, we get that question a lot. Of course, there's a potential. Of course, this is a de novo sequence, so it doesn't exist in nature.
Yeah.
So potentially, human can or animal can engage immunogenic reactivity with this. Well, we can partly address it in silico these days. Doing the running of these in silico prediction, whether or not it's a strong T cell epitope or not. So we can eliminate this if we need. But other than that, of course, you just have to do it.
Yes.
You never know what's going to happen. So yes, you're right. There's a potential that it can be highly immunogenic.
Okay, thank you.
So, Jun, that was really great. So I'm really impressed how versatile peptides can be when they're cyclized. So I think for each of the examples, you showed just one peptide of many that you may have selected. And so how general is this? Because maybe you got 10 peptides, for each target. How general was this activity?
Thank you. Great question. So of course, it depends on the cases. But let's say through this rapid system selection, we would get, say, on average, 10 good-looking sequences, independent, seemingly independent class of binding sequences. And then we test these first grafting compatibility, meaning that whether or not you can express these protein, okay, and whether or not these expressed inserted protein can bind to the target. So then the success rate, roughly, again, it depends on the cases, but its success rate is about more than 50%. Like, let's say out of 10, we can get 6 successful grafting working or compatible sequences. And then, of course, then you have to test if your intention was to get the agonist or antagonist, whether or not these have such biological activity. Then, of course, it really depends. Because after the selection, we have no idea which domain these peptides would bind, right? It could bind to the key place, or it could bind to the base of the protein, which is not really accessible to this kind of format. So that needs to be tested one by one. So overall, but I can say just this. So we have the receptors, sort of, the target molecule that we aimed for. In 100% cases, we can have grafted mirror bodies. That's recognized cell surface molecule. That's the overall success rate. Like I said, some peptide didn't work and some worked. But overall, in terms of getting a binding, good binding mirror bodies, the success rate is 100%. That's about it.
Right, but so you, for example, showed that you could get agonists. So how many, if you tested 5 peptides, out of how many of those peptides were you able to get an agonist?
Oh, so for these one that I talked today were already published sequences. So these peptides were not selected for this purpose.
Right, right.
We already had that.
Yeah.
And then for the other project that we do currently, if our intention was to get the agonist, for the toughest agonist so far, we had to repeat this selection cycle like 6-7 times until we get this activating compatible peptide sequence. So it's just, how badly you want that.
Okay. So, and out of each cycle, you're getting 10 peptides, maybe?
Depending on the target.
Yeah.
Sometimes we get just 2 or something. Sometimes we get 30.
Okay.
It's really variable, yes.
All right. And so thus far with structures, do you know how many different types of surfaces these peptides bind to? How broadly distributed they are over surfaces, and are there any, do they like pockets, or are there any rules so far?
Yeah, so the thing is, we haven't been able to solve the structure of too many, pairs.
Right.
So we don't have that elaborate information.
Yeah. Well, I'm sure there's somebody who can predict where they bind, anyhow. All right.
Yeah, we did that, and then it didn't work that with that program.
Right. Okay, thanks a lot.
Hi, thank you for a beautiful talk. So my question is about the diversity of the peptide binders you have tested. So the first question is about the length diversity. So have you tested, different lengths and have noticed how tolerant it is in this kind of insertions?
The standard library consists of the peptide lengths of 14 to 17. That's like a standard, regular library that we use.
Yeah.
And sometimes we push forward to a 20 or 22 even.
Okay.
But it's not necessarily longer the better, because if you have longer sequence, you have less diversity.
All right, thank you. And the second question is about chemical diversity, I guess. So I noticed you included cysteines on the 2 ends of these peptides, so I assume those would form disulfide bond. So have you tried, including additional cysteines in those peptides so that they can form multiple circles?
Thank you.
Yeah.
Yeah, some of you noticed that some of them have cysteine. Yeah, in certain cases, it's essential to have a cysteine disulfide at the base, but it's not always the case. So we still don't know. Well, that's probably because of how strong or how well this peptide moiety is optimized for assuming certain conformation. If it's autonomously folded in a small module without having a bond, then you don't need this cysteine.
Yeah. So have you tried inserting cysteine in the middle so that they can form, additional kind of circle
Right.
structure that can accommodate
Right.
different surfaces?
Short answer is no, because when we have this rapid peptide selection, sometimes we have cysteines appearing at the middle, and then that will make short peptide. I mean the small ring peptide, what we call lariat shape molecule, which is not compatible with this grafting of the macrocyclic peptide. So we don't do that.
Okay, thank you.
Thank you.
Final 2 questions, please.
Thank you for this inspiring talk. I might have a naive question at this point, but did you ever try to use immune cells to do drug delivery for your mirror bodies? So, for example, having a T regulatory cell, having a T cell with an sign-notch receptor that, when it arrives at a tumor site, for example, would then deliver the peptide of interest, because it's a lot of delivery about cyclic peptides.
Yes, good question. So, of course, the drug delivery, particularly the protein delivery to one particular tissue or cell or organ, is one of the greatest applications that we think of. We don't have a good case to make here at this moment, but that's definitely the direction that we are chasing after. Thank you.
Thank you very much.
And the final question.
Do you think these peptides are less flexible than the Fab arms on an antibody, or more flexible, for instance?
You mean the flexibility of the Fab arm itself?
Yeah.
Uh,
How do you think they would
that's a difficult question, depending on which point you think that the hinge point is. If it's a Fab arm from the classical hinge point, it's this movement, right? Compared to that, the conformational freedom of the peptide is much smaller. But still, it's not restricted in a way. So this peptide or loop position can flip around like this. So in terms of the angles, probably it's not that different. Of course, depending on a particular sequence, it should have different freedom. But it's difficult to say, I would say.
Thank you.
Thank you very much.
Thank you.